with you all today. I think everyone's already familiar with a GAMP deficiency, so I'm not going to talk about this slide here. So for the families and for the parents, I want to make sure that you all have a sense of what the carrier of the gene is, whether it's for the creatine transporter deficiency or it's for guanidinoacetate methyltransferase deficiency. So what people are doing around the world at present is trying to find ways to deliver genetic material to cells, whether that be by synthetic methods, like lipid nanoparticles, like for those of you who received the COVID vaccination, that was a lipid nanoparticle-based delivery system with RNA, a genetic component, or altering a virus that is presumably evolved with all of us over the millennia that has already optimized its way of delivering its genetic material to our cells. So in this present time, in 2024, most of the technology is in altering viruses. And one of the most common viruses that's used for this is something called adeno-associated virus. And this is a member of a family of viruses called Parvoviridae. So if you have a dog and the dog gets a parvovirus vaccination, this is in that same general class. It's a small virus, but it's a very commonly used virus because of its advantages. Essentially, with minimal exception, there's no disease in humans associated with this virus. That's the main advantage. Essentially, that's correct. The disadvantage is that the genetic material it can hold is small. For these disorders we're talking about here, these three in the creatine deficiency family, that won't matter because the genetic material that needs to be delivered is small enough. But for other disorders, muscular dystrophy, cystic fibrosis, hemophilia A, that genetic material takes up almost all of what the virus can hold. So that's one of the big disadvantages. But again, for the disorders we're talking about here today, it's not really a disadvantage, again, because the genes are smaller. So with these viruses, this is the wild type virus, a representation of the genetic material. These genes, the replication genes, and the capsid, or the coding material of the virus, the protein that carries the genetic material, are removed, and you're just left with these packaging elements at the end. And then essentially you put in what you call an expression cassette. You put in some genetic material that turns on the gene, some genetic material that stabilizes the gene, and you put the gene itself, what we call an expression cassette. So whether it's us, or it's Laura Baroncelli, or it's the Walia group, we're all developing our own expression cassette based on different things that we think are important, with something we call a promoter that turns the gene on, something we call the transgene, in this case, for me, guanidinoacetate methyltransferase genetic material, a polyadenylation signal to stabilize, simple terms, and then these packaging elements at the end. So that's what we're doing. We're taking a virus that's evolved with all of us, that knows how to infect or transduce our cells, and we're changing it to deliver genetic material that's important to treat these disorders. So I just want to make sure that's clear so you understand what we're starting with and where we're going, okay? So everyone's familiar with this, a pathway already, but in the past, we obtained the guanidinoacetate methyltransferase deficient mice, which were developed in Germany about 20 years ago. These mice biochemically have high guanidinoacetic acid and low creatine, but their behavior is almost normal. The group that studied and developed these mice in Germany found very few deficits, very few, and the ones they found were very small, minor. Our group, through UCLA, a group that we work with, studied the mice and was able to find some additional deficits that were more helpful to study what the therapeutic response would be to our gene therapy. So as was mentioned already a little bit about a biomarker for the creatine transporter deficiency, we've actually used some of the same technology to look at a biomarker for GAMP deficiency, and that is PET-CT scans, positron emission tomography, computed axial tomography scanning. Now, if you've come across this in the past, it would probably be in the situation where you heard of someone having cancer and they were trying to figure out if they have the cancer elsewhere in their body. They're given a radioactive substance, short-lived radioactive substance linked to glucose or sugar. You give that to someone, it tends to go to cells that are rapidly either dividing or using a lot of energy, and it goes to those locations and you can visualize it. In this case, we've done this with the GAMP deficient mice and wild-type mice. So here's the head. We're looking in profile, the head of an adult mouse, and the brain is in the circle, and we see these different colors. Red is more activity, blue is darker activity. So here's a wild-type mouse. Here's a mutant mouse. You see much less brain activity, energetic activity in the brain. And then with our gene therapy that I'm going to tell you more about in a moment, we see this improvement in brain activity, in utilization of glucose in the brain. If we look quantitatively here, here's our wild-type mice, our mutant untreated mice. There's less glucose activity in the brain, but then we see some restoration in brain activity with the gene therapy that we developed for GAMP deficiency. We've also compared these groups. We've compared it with creatine treatment alone and gene therapy, and in fact, the gene therapy is better than if we do creatine supplementation. So this is one of the studies that the group at UCLA found, the group that we work with at UCLA found is different in GAMP deficient mice compared to wild types that the German group didn't study at the time. It may not have actually existed back then. But it's something called the Barnes maze, and it kind of looks like what we have here in this picture. Essentially, mice kind of don't like to be out in the open. They think they feel vulnerable, so they like to go in the corner of a cage or like to hide. So what this is you have this device in a room. You don't move it around, you keep it in the same position, okay? And there's these holes that the mouse can go in, and then there's this chamber here. And the mouse generally will want to try to find this chamber to hide in. So mice can learn to do this over time. You release the mouse, and you time this, and the mice will travel in a different way to find that spot. So you have a period of training of these mice where they learn this on a daily basis, 4 days in a row. And then after a day, you test them again at 24 hours. And then you wait another week or 5 days, and you test them again to test short-term and long-term memory. So it's a good test to try to understand if the mice are learning. It's a really good test for it. Again, a test that we found there were differences between the GAMP mice, GAMP deficient mice, and wild-type mice. So this is the patterns that you typically see with mice. So you can have this random pattern. Typically at the beginning, it's learning where that spot is. That male or female mouse is learning where that spot is. And over time, they may do a serial tracking where they go from hole to hole to hole, or eventually they'll remember where it is and go directly to it. I hope this makes sense to everyone here. We need methods to objectively determine if the treatment is effective. We can't just look in the cage and say, those mice look better. It's not objective. We have to have, just like in a clinical trial, what you're doing in the core outcome set is you're trying to find objective measures so that in the end, you can apply to the FDA in this country and say, we have objective measures that show benefit. So then they approve the drug and the insurance companies have to pay for it. Because otherwise, if you don't have objective measures, how do you know that you're really providing a benefit? So we have to do things in a way that are blinded, that whoever's doing this test doesn't know what group the mice are in. Are they wild type? Are they treated? Are they GAMP deficient? They have to be blinded, and they have to do it objectively. So when we take the mice, these are wild type mice in this column. This is day 1, this is day 2, this is day 3, this is day 4. These are the wild type mice. So we have a period of random searching over time. We have this direct where they find the hole. We have this serial. And over time, the wild type mice go from kind of decreasing the randomness to increasing more of the direct or the serial. You see that the gray is disappearing in this first column. But if we look in the GAMPed mice, we see that over time, they learn some, the gray decreases, but not nearly as much as the wild-type mice over here. This is the GAMP deficient, this is the wild-type. When we have the treatment, this is the GAMP deficient here, and this is the wild-type. When we have the treatment, we see that we have this improvement. But what's the best part is if we look over here, if we wait 24 hours, we see that here's the wild-type is all direct or serial. The treated mice show much more direct compared to the deficient mice, that is in our gene therapy treated mice. But when we go to a week later, the mice that were not treated with the red arrow have lost that. They lose this direct spatial method of learning, of memory, whereas the treated mice retain that. So we have an objective measure that shows that the gene therapy, when given, actually stays with the mice. They learn, and the learning appears to be permanent, at least over a week's period of time. So we have this objective measure that showed that the gene therapy that we were doing, that I'm going to tell you more about in a second, does work. So the method that we have is actually intravenous injection. In a vein in the tail can be given, in a human it can be given in the arm. And what we saw in those initial studies that I described to some of you that are here today that were here 2 years ago was that we saw that if we compare our groups, this is our wild type group, this is our knockout group. So in the liver, if we look at creatine, we have almost no creatine in the knockout mice. But in the treated mice, the creatine greatly improves, very similar to wild-type mice. We see the same in the brain. In the brain of these mice, the GAMP-deficient mice, untreated, have essentially no creatine in our hands. And in wild-type, it has this level. And in the treated, it's very, very similar. There's not a statistically significant difference. With guanidinoacetic acid, we see a similar but inverted pattern. The guanidinoacetic acid in the brain is very low. In the wild-type mice, it's high in mice that are not treated. In the liver, in the liver. It's high in the mice that are not treated, but it's normalized. Now, in those studies that I presented here about 2 years ago, the concern that I had at the time is we have this persistent level of GAA in the brain. So again, here's the wild-type mice, looking at GAA in the brain. Here's the untreated GAMP-deficient mice, but here is our treated mice. It did not lower as much as we would have liked to see it lower. So subsequently, since this time, we've re-engineered our virus and tested multiple viruses to ultimately end up with one where this is now resolved. So we've been spending a lot of time trying to understand what is the brain dysfunction in guanidinoacetate methyltransferase deficiency. What's happening? What leads to brain injury? What leads to these abnormalities? And we have some hypotheses that we're trying to prove and other areas that we have been able to demonstrate it seem to be correct. And I will tell you that I entered this field from the arginase deficiency field, what Nicola brought up on the first day. I actually was studying arginase deficiency where you have hyperarginemia, elevated arginine. And at the time, I knew that there were many guanidino compounds produced. GAA is one guanidino compound. In arginase deficiency, there's about 13 or so. And I said, I think I can study GAMP deficiency to give me a clue as to what's going on in arginase deficiency. Now, I didn't know at the other time that some other people had a similar thought. I think Nicola had this thought, and Andreas Scholze from the video yesterday also has had this thought independently, because Andreas and I have spoken about this. So we've come up with this hypothesis as why do GAMPed children and adults have seizures? I think it's somewhat unknown as to why, and I think Matt suggested something the other day. He said, Matt Skelton said, an alteration, I think he was talking about creatine deficiency disorders, but he said an alteration or an imbalance in excitation and inhibition So when we think of a seizure disorder, there may just be too much excitation and not enough inhibition. And what I hypothesize is happening in GAMPed people, and probably mice, is this. And there is basis for this, and we're trying to prove this. There's basis for this in the alcohol literature that I'll just tell you about in a second. So what you typically have in this, what I'm showing you in this diagram, is here's a neuron, okay, a neuron in the brain, and it releases a neurotransmitter called GABA. It's the main neurotransmitter in the brain. And it goes across the synapse and it binds with these receptors called GABA-A receptors. Now, GAA, we have found, and I think others possibly in the past, have found that GAA acts like GABA. It's a mimetic of GABA. It works on these receptors. Now, normally, none of us in this room, if you don't have GAMPed, you really don't have GAA in your blood. It's very, very low. It's very small level. But when you have GAMP deficiency, you have this. You have it in your blood. So what happens is, probably, what we hypothesize is the receptors for GABA, they become internalized in the cells. They don't disappear, but they get internalized. So in this diagram I show here, when you have GAA around, it causes internalization of these receptors. And probably this leads to this, then you have less response to inhibition. So if something changes physiologically for that patient, you'll have a seizure. Now what we think can happen is if you can reduce the GAA enough, then these receptors will come back and it will restore that imbalance of excitation and inhibition. It will bring the inhibition back up to eliminate the seizures. That's what we think will happen. That's what we're trying to demonstrate now. Now, you might say, where did you come up with this crazy idea? Well, I mentioned the alcohol literature. For those of you that are physicians, you know that if someone takes too much opiates and you send them to a rehab center to withdraw from opiates, it's generally not life-threatening. But someone who's continuously inebriated with alcohol and you withdraw alcohol, it is life-threatening for them. And they have to be treated in an environment where they're treated with benzodiazepines like diazepam, other agents like this, because there's an imbalance, because the receptors become internalized. So I think that's what's happening here that's causing the seizures. And again, we're trying to demonstrate this. So again, I bring this up because we're trying to not only develop the therapy, but we're trying to understand what's going on in the brain and how it's going to respond to the therapy. So here's an example of the alteration in the inhibition that occurs in neurons. If we take GABA, which I mentioned is the main inhibitory neurotransmitter in the brain, and we take a mouse, this is a little bit gruesome, okay? You take a mouse, you anesthetize it deeply, very deeply, and you take out its brain. And then you take a certain section of the brain and you place it on a stage under a microscope here, and you put electrodes into a neuron, a recording electron, a stimulating electrode, excuse me, a recording electrode and a stimulating electrode, and then you add to the bath the liquid that's supporting this brain, which is basically an artificial cerebrospinal fluid, and you add GABA, again, the main neurotransmitter, main inhibitory neurotransmitter, you'll see an alteration as you increase the dose of GABA. You'll see that I think everyone can see how this declines. It's inhibitory. If we do the same with GAA instead of GABA, we see the same kind of response. So we know, and this agent here reverses it at the end, which acts on GABA receptors. So we know that GAA acts through this GABA mechanism. We know that's how it works. It turns out, as I mentioned, there's more similarities between arginase deficiency and GAMP deficiency than I realized. And I can't explain to you why the symptoms in the patients do not overlap. I can't explain it. I don't understand why. But it turns out, at least in the mouse model, there's more than just GAA in the brain. There's multiple other guanidino compounds in the brain. There's guanidinopropionic acid, there's guanidinobutyric acid, there's argininic acid, there's guanidinoacetic acid, all in the brain of GAMP deficient mice. In humans, we don't know. I don't know that we'll ever know because there probably aren't or ever will be really a lot of autopsy specimens to look at. But I think getting at this understanding of what's going on is important to know if the therapy is effective, because we can measure these compounds in the brain of the mice. So now I'm going to talk to you a little bit about the new studies we've been doing to reduce the GAA in the brain. So this is the timeline of what we've been doing. So mice are treated when they're 2 months old, and we have male and female GAMP deficient mice, male and female wild-type mice for all of these studies in equal numbers. So the mice are 2 months old. We collect blood on a monthly basis. We have the intravenous adeno-associated viral injections at 2 months, and we're following the mice this time for 6 months. Our previous study, we followed them for a year. This time we're only doing it for 6 months. We did doing some studies at 2 months in one cohort of mice with all the different viral vectors, and other mice go to 6 months. We collect blood at all these time points. We're collecting urine at the final time point. We're analyzing the tissue like the brain and the liver for these compounds like I've mentioned to you already. We assess them daily for weight. Weight in mice is a good surrogate of health. When mice start to lose weight, usually they're not healthy, unless they're very aged, but these mice are not aged. In the blood, we have monthly collection for analyzing guanidine, acetic acid, and creatine. Now, importantly, 2 weeks after the injection of the viruses, we take the mice off creatine. So typical mouse chow has fish meal in it, protein derived from animals. That will have creatine in it. So after 2 weeks, we take them off all creatine. So all the data you're going to see after time point 0, the mice are getting no creatine supplementation, no creatine in their diet of any form. They're on a vegetarian mouse chow. And this is regular mouse chow. This is what the vegetarian mouse chow looks like. So we developed 4 different types of these viruses that have different components. The details aren't too important, but I'll just explain them briefly. We have 2 of the different types of viruses that have a liver regulatory element. That is, the gene, GAMP, is only expressed in the liver. We have another one called the CAG promoter, which is really about 90% of all clinical trials use this promoter in people, which expresses ubiquitously, expresses generally everywhere, even though most of it really ends up in the liver. And then we have a fourth vector that also has that promoter. And then what we did is, in 3 of these viruses, we have the GAMP gene we've been using before, but then here we changed the GAMP gene a little bit. We did something called CPG reduction. We did this to try to have the gene not well recognized by the immune system, because there can be components of genes that can be recognized as foreign. And we tried to alter the genome itself, the gene GAMP, so that we would reduce this. And then we took what we call different serotypes, what you might think of for colds or other things, strains of the virus, that ones that we are used in human clinical trials already, that we thought might be better, might be worse, in some way to assess what we might use in people. So we used one called RH10. That's the one we used previously. We redid the data. We did these studies again. One called AAV8, AAV9, and again AAV9. Now, AAV9 is a common one used today. There's a drug called Zolgensma for spinal muscular atrophy, a horrible disease that affects children. They typically die before they're 2 years old. And it's the largest gene therapy administered drug in the entire world. Over 3,000 children have received AAV9 already. So we know, and there probably isn't that big of a difference between the serotypes regarding safety, we know that there's many people been treated with this. It's in a lot of ways, unfortunately, the biggest clinical trial there's going to be out there, because it's not a clinical trial, it's an approved drug. But we don't really know what's going to happen long term. Those girls and boys that have spinal muscular atrophy are really going to tell us over the next decades. But we treated our mice with these different viral vectors. And, oh, I wanted to go back one slide. I forgot to mention, as I touched upon it briefly, we have the short-term study. And again, this is just intravenous, so a cartoon of a child in a bed getting it in their arm. It's not in the brain, it's in the arm, okay, in the vein in the arm. Could be the vein in the leg, could be a vein where you ever want to pick it to be. But it's typically vein in the arm, in this case, the tail vein. And then we have these long-term studies. So we took the mice, we injected them with the different viruses, we collected blood, got the clear liquid called the plasma, and we analyzed what the response was. So In blue is time point 0, in red is time point 2 months, and if you look at the wild type, we look at creatine, it's about the same between that time. You wouldn't expect there to be a change. We're just injecting them with saline, a vehicle. The untreated mice have very low creatine. The GAMP-deficient mice have essentially no creatine. You wouldn't expect there to be much of a change there. But if we look in our RH10 virus, it goes up a lot. If we look at our AAV9 virus, it goes up a lot. In the one we tried to re-engineer the genetic material, it didn't go up that much. And then if we look at guanidinoacetic acid, we want to see resolution. Of course, we want to see it go down. So in the wild type, it's the same. In the untreated, yeah, it's down, but I don't really know why. I think there's just some variability in the testing, but there really is no reason for it to be down. But if we look at all the viral vectors, the standard, where we had the standard genetic material, they all go down, except the one here where we had the altered genetic material. So we decided that going forward, this one where we tried to decrease these CPG domains is probably not the one to proceed with at the present time. Now we looked at the genetic material in the liver, the protein that's there, the GAMPed protein itself, and I think it's easily seen here. This is the wild type amount of protein that's in the liver. This is the knockout. You really don't see any in the mutant mice. And then you see increases in the amount. This is the RH10 vector, this is the AAV8 vector, and this is the vector that I called AAV9 with the standard GAMPed gene. Has the highest amount of expression in the liver. And then if we look at the livers from these mice, sections of the liver, and we stain it for the GAMPed protein, which will be in brown or a little bit orange here. If we look at untreated mice, it's just this blue background, there's none. This is human liver. This is a normal patient who had some liver, or had a liver disease and took a piece of liver and we stained it for GAMPed, and you see all the brown here. If we look at our different viruses, here's the RH10, you see the brown, orange out here. In the AAV8, you see the orange here as the amount of protein. If we look at this virus, the AAV9, you see it spread around also throughout these liver cells. But more importantly is what's going on in the brain, because that's what I told you I was concerned about from the studies we did 2 years ago. Now, there's a lot on here, so I'll try to just point out the salient points here, is if we look at the creatine in the brain, in the GAMPed deficient mice, with no treatment, there's very little. Here's the wild type, the normal mice, males and females. And then we see improvement in all of these. I'll point out this one is the one that I call AAV9 here, okay? Now, if we look at guanidinoacetic acid, if we look at the GAMPed deficient untreated, here's the level. In the tissue of the brain, here's wild type. As I mentioned, there's very little there. And then if we look, we see this reduction of GAA in the brain with our 4 viruses, but the AAV9 one is the best. And then I mentioned that other guanidino compounds exist in the brain of GAMPed deficient mice, one called guanidinobutyric acid, and here it is in that AAV9 mice, the treated group. It's essentially non-existent. So it appears in our assessment that AAV9 with the standard GAMPed gene, we can resolve the GAA and we can resolve at least one of these other guanidino compounds, guanidinobutyric acid. Now, I mentioned that we're continuing this study to 6 months, and if we look here, again, weight being a surrogate of health, in black, female mice, male mice. In black are the untreated mice. In blue are the wild type. And then these are the 3 different viruses. They all gain weight in females. And you see the same pattern in the males. You see this improvement in weight. Again, surrogate of health. And then looking in the plasma on what's happening here also, looking at the creatine levels. So here's the untreated mice. In the plasma, it's essentially 0. With treatment, the creatine improves in the female mice. It also improves in all the male mice of the different viruses. Again, leaving out that specialized genetic changes to GAMPed, which we've abandoned. Same if we look at GAA. Here, the untreated mice, it's high, about 50 micromolar. It's essentially undetectable in the mice that are wild type. And in treatment, we see this marked improvement. We see the same pattern in the males. In the urine, it's also very interesting. You see the resolution. So creatine is very low in GAMP deficiency in the urine. Here it is in normal mice, and we see this resolution with these viral vectors, particularly the AAV9 again here. And same with GAA. It's very high in untreated mice. It's non-existent in wild-type mice. It's non-existent with our viral vectors that I've shown you here. So to conclude, in the development of an peripheral intravenous effective gene therapy for GAMP deficiency, we're continuing to advance this approach. We have some additional studies that we want to do. In these long-term mice, the 6-month mice, we want to determine the levels in the liver, creatine and GAA in the tissue itself. We were taking out the brain to examine certain regions for creatine, GAA, and guanidinobutyric acid, that other guanidino compound I mentioned. We want to determine the GAMP protein levels, those spots that I showed you on there. We want to determine what that's like at 6 months also. And we want to look in the other organs and see where we have some expression of GAMP protein. With that said, these studies really would not have been possible without two groups. The first is ACD. ACD has provided us $20,000. You all have provided us $20,000, two $10,000 increments over the past 2 years to get these studies going to the advancement stage. The second is Andreas Schulze and Alona Tekiova at Toronto Children's, at Sick Children's Hospital in Toronto have really been our partners, and we would not have been able to do these types of metabolic studies without them to show you the efficacy today of this therapy. So I want to recognize them, and again, to recognize ACD, and then of course the members of the group at UCLA who've been working on this with me for the past several years. Thank you.
Very cool data, Jerry. So, anyone have questions? Oh, yeah, Heidi.
Thank you for that energetic and very educational talk. You've set the bar for the rest of the afternoon. I had a question on slides 9 and 10, and I hope I can remember both of them. I want to make sure that you didn't spell it out, but I assumed that when the folks going through withdrawal from alcohol and they're taking the benzos, and the benzos help with the GABA being pulled back, that you were going to say then the GABA was restored and everything was better, and right?
The seizures would be gone over time.
Okay.
Yes, that's right.
Okay, I just want to make sure I got that right.
They
and
would not
then
need
there was something on the next slide, sorry. So the thought, okay, so you have this model of this happening and that the GAA is acting similar to GABA. Is there another incidence that we now say, okay, so when the GAA acts like GABA, we know that the GABA pulls back? Is there another system where that happens so we can make the leap from this to that? Is that
another system where elevated GABA causes the problem on the last slide? Yes. Yeah. Yes, there is. In patients, there's a disorder, another rare disorder called succinate semialdehyde dehydrogenase deficiency, which has an overabundance of GABA, and in those patients on imaging show reduced GABA-A receptors.
Interesting.
In people.
Okay, awesome, thank you.
Yes.
So you said that they show reduced GABA on imaging?
They show reduced GABA-A receptors on imaging.
What type of imaging?
PET-CT.
And so you would conceivably do similar types of tests on...
Correct.
Yeah. And is there a way also to, we're talking about this internalization happening at the synaptic cleft, right? So is there a way for us, and of course we don't know what type of GABA receptors, what subtypes of GABA receptors is happening. I think in your last talk you referred to extrasynaptic being involved. So do you think there's some staining of the cleft that we can do and or electrophysiology with and without GABA agonists, and try to understand what are the specific subtypes of GABA receptors and prove out this hypothesis further?
Yes, I'll say that I didn't want to get into the weeds of it And that's why I didn't specifically talk about this, but this will make you believe in a divine intervention. These receptors, it's unbelievable. These what are called GABA receptors, there's more than a dozen different subtypes throughout the brain. And it makes you wonder, how did we get here through evolution? The complexity of these receptors. And what Sangeetha is talking about is, and I oversimplified it. I made it this way. But I think, see, normally this area is somewhat sealed. It's called the synaptic cleft. And the concentration of these neurotransmitters that are released are really high concentration. But it's just there for a very brief period of time to go across this extremely short distance and stimulate these receptors. But as you could imagine, if you have normally in someone essentially no guanidinoacetic acid, or very, very tiny amount, that very tiny amount maybe isn't in the cleft, but it's around here. It's in this location more, if I may say that. Extrasynaptic, outside of the synapse, and it provides some regulation of activity of this itself, an additional regulatory mechanism. And we believe that there are extrasynaptic outside of this that are in this location that are affected by the guanidino compounds. That's what we think is happening, actually, and we are looking at some of these GABA receptor subtypes to see if there's a difference. If we take the GABA receptor subtype knockout for certain subunit, and compare with regular GABA, normal mice, normal GABA A receptors, and looking at the differences. So I do think so. Unfortunately, it's not as clean as we'd like it to be, but there does appear to be a little bit of, at least a little bit of a difference in some of the guanidino compounds. It's a little early to say, but hopefully by the end of the year.
Thank you.
Yeah, and while I'm walking over, I didn't know it, but I just looked it up. Gabazine is very selective for GABA A, so that suggests that you can reverse the compound, or
Yes.
reverse the GAA effect with antagonizing selectively GABA A.
Exactly right,
Yeah.
right here. Yes, exactly. Coming back up to baseline, right.
So you mentioned that you weren't going to continue with the one form of the AAV9, but what are your next steps? Can you speak a little bit more about your next steps towards clinical trials, and are you going to move forward with the AV8 and the AV9?
Yes, so we're moving forward with this one. We're moving forward with the AAV9 with this what's called the very commonly used regulatory element called chicken beta actin promoter, CMV enhancer. We are fortunate enough to have obtained some substantial funding recently to start the greater efficacy and toxicology studies in mice. So that's what we're taking forward. We can only take forward one, and I don't think there's any reason to take forward others, because the process is long and complicated in the US and I think other countries, and there's reasons for that are good, but there's no reason to try to develop two. There's only reason to develop one, and that's what we're taking forward.
Okay.
Sorry, I might have missed this earlier, but in your CPG deleted AAV9, did you delete the CPGs just in the transgene or in the whole ITRs as well?
Only in the transgene.
Okay. And do you have any idea why you think there's decreased expression, or just some misfolding or something, or lack of codons?
That's what I guess too. I don't know. I have to say, because it's like that, we're just not pursuing it anymore.
Yeah,
we're not going to
fair.
investigate it.
Fair
Yeah.
enough. Thanks.
Thank you.
Okay, so GABA transporter is in the same SLC6 family, but for the receptors, this is a little bit different with the GPCRs. However, there are some other kind of corresponding receptors that are also have transporters in that same family. Have you looked outside of GABA with any of like the neurotransmission? Because there are some specifically that kind of sit just outside that synaptic cleft, like kind of down the sides where you were pointing. So have you looked at any of the other really common like receptors in neurotransmission to see if those are also impacted at all?
We haven't. We've only looked at certain GABA subtypes. To be honest about it, We wanted to do that, and I shared this with Matt yesterday. We've submitted a grant on this 7 times to the NIH, and I thought it was the best grant I ever wrote, and we could never get it funded. And so we don't have the resources. You know, the person that I have that does electrophysiology, essentially, you know, the benefits in California are very high, and I pay essentially $20,000 a month just for her to do these studies. It's just too expensive. We can't do it unfunded.
Gotcha.
We'd
Thank
like
you.
to, but we can't.
Okay, so I'll ask one personally interested question. Do you know the residual GAA that you have, is that in whole brain lysate? Or is that measured in the CSF?
No, it's in whole brain.
It's
What
whole
we
brain
did
lysate?
have is whole brain.
Do you
Yeah,
No,
whole
go ahead.
brain. It's, you know, again, I can't thank Andreas and Alona enough from Sick Kids, but we send them milligram amounts of brain that we weigh in a tube that's frozen in liquid nitrogen and dry ice, and they do the analysis. It's whole brain, it's not CSF.
And so it's still a very small amount, which is
Yes.
amazing. And then do you think that that is intracellular, or do you think it is actually extracellular
Yeah,
GAA?
it's a great question. We do perfuse the mice before with saline, but I don't know how much that enters really into that space.
Okay.
What I mean by that, to people in the audience who might not have experience with this, after the mice are euthanized, we put a needle in the heart and we flush fluid through to rinse out all the blood out. So we know that, you know, if the blood's contaminated with GAA and some is in the blood vessels of the brain, and you're just giving someone a piece of the brain, well, they're not going to only detect what's in the brain, they're going to detect what's in the blood vessels. So we flush it out with saline so that's not, the chance of it remaining in the blood vessels is very small. What's there is what's in the brain. So I don't know, but we did, yeah, I just don't know. I don't know.
Well, I think it speaks to, I think it was Jagdeep's question earlier, which is how little GAA do you need for a good clinical outcome or to say it's safe? And if the mechanism of toxicity is what you're positing, which is agonism of the GABA A receptor, if it's intracellular, you could probably get away with a little bit more than extracellular. But yeah.
Presumably, yes.
Yeah. No, it's beautiful data. And I think
Thank
together that was an awesome collection of gene therapy studies.
Thank
Yeah.
you. Just end my slides here.